Great. So now we would like to move to Dr. Gahan Pandina from Janssen. Gahan, we'll turn it over to you.
Thank you very much, Linda, and thank you to the organizing committee of the investigator meeting today for inviting me. Can everyone see my slides?
Yes. You'll put them in presenter mode?
Yes, I will.
Okay, perfect.
Wonderful. So I'm here to give you a bit of a journey, a different kind of talk, hopefully inspirational, leading you from research that, as Dr. Heinzen was outlining, has gone on for many years, and a lot of thinking gone into the development of novel research in early psychosis. So I just am mentioning here that my disclosures: I'm a full-time employee at Janssen. I work in Janssen as in drug development in psychiatry. My background: I'm a clinical psychologist by training with a background in neuropsychology, and the views I'm going to be talking about today are my views, not those of Janssen or J&J. So what will I say? Well, first I'll take a little bit of a look back at where we've been and the journey for me and from my perspective as well. I'll talk a little bit about psychosis research and how we're reaching a new maturity in the approach that we're taking to looking at psychosis and related symptoms, and then talk a bit about AMP, although I think the team today has given you a wonderful overview of the research that's planned, the endpoints that are being examined, and the impacts that those endpoints may have in research already that we can expand upon. So when I started looking back at when I first was involved with many of the researchers in this group, there was the International Prodrome Research Network meetings that used to be held on the West Coast. They were often hosted by the Staglin family and Ty Cannon. And one of the first talks that I gave in this network was in 2003. And actually a paper came out shortly thereafter that Barbara Kornblatt, who I saw on the call earlier, was a champion of, and others. The challenge then that we were discussing was prodrome, the prodrome in schizophrenia and first episode. Much of what my talk focused on then was prodrome to what? How can we identify early illness factors, as we were talking about them then, what prior to first episode or around that time? What can neurobiology or neuroimaging research tell us? What factors predict conversion to full diagnosis at the time? Can we track the course of illness and potentially target symptoms in this early process? And could we identify mediators and moderators? And then, of course, is the prodrome a state in and of itself or a precursor? So as I was thinking about this, many of the questions that we had at that time are still around today. That is, we're still trying to find the answers that we were looking for even way back at that time. And in part, this was the state of the science shortly thereafter in biomarkers. At the time, we were looking at, and this is a general discussion of precision medicine that was published in 2005 about biomarkers in disease, drug discovery, and development. And typically, biomarkers were used to measure target engagement. We use PET receptor occupancy, for example, to make sure that our drug is triggering in the places in the brain that we think it should be, and it's brain penetrant. We look at both PD effects and treatment responses using things like EEG. These have been sort of standard processes that we've done in neuroscience for many, many years. And then we've maybe used these markers for population stratification in phase 2 and phase 3. But you can see there's sort of a logical progression here. And other disease areas, including neurology, cardiovascular disease, oncology, have benefited from these sort of standard approaches. These haven't been as robustly successful in neuroscience, in part because the endpoints that we're looking for and looking at are very much more challenging and complex in some ways. And also, we've relied on many, many subjective measures to measure outcome. What about biomarkers in 2022? Well, we've had a total revolution in how we think about the use of biomarkers. And these, while some of the same candidate biomarkers or candidate tools that we use are still around, and we've refined the ability to look at that data, to analyze them, to process large databases like neuroimaging. We've also increased our sophistication in looking at fiber tracts in the brain, pathways. We've also had a revolution in genetics and proteomics, as well as fluid biomarkers. And that, in addition to our ability to do things like momentary assessment, assess behavior through wearables and trackers, track behaviors across a wide range of platforms, all of those things are revolutionizing the way we think about biomarkers in 2022. And of course, consortia and partnership amongst companies, CROs, and other research partners, academia, clinical sites, and foundations, and regulatory agencies are critical here. How do we utilize the data that's coming out of these to think more maturely about what type of indication might we develop for a novel therapeutic? So I would argue that there's never been a more exciting time in psychosis research than right now. We are just poised on the brink of many, many applications of novel science that's been exploding over the past 10 years. With the development of these large data sets is going to lead us to answer much more mature and sophisticated questions. So I'd like to take you a little bit on a journey of why I think that. First of all, we've developed robust clinical outcome assessments. We started this day talking about the psychs, which is a combination reformulation of the CARMs and the CYPs, a marriage of those two. I think the sophistication with which we've begun to look at clinical symptoms in these clinical high-risk populations has been a critical factor in our ability to entertain this type of research and to utilize these potentially in a regulatory setting. But CHR clinical trajectories are not easily predicted. We know this from many different areas of research. I've just pulled out one example here. This is from the NeuroPro trial, where the investigators looked at trajectories of ultra high risk participants over 30 months and tried to identify trajectories and predict those trajectories based on different outcome measures. And there were several factors that were predicting different trajectories. For example, female gender and substance abuse were predictive of attenuated positive symptoms in depression. Age was also predictive of depressive symptoms, and cognition was predictive of high negative symptoms. But as you can see, these models, are challenging. And in fact, we're looking over a brief period of time to see whether or not we can predict clinical symptoms. However, we're improving on this risk trajectory prediction. So this is another example using the Naples cohort replication. And in this analysis, the investigators used a 3-group model to identify in the Naples 2 cohort and then replicate in Naples 1 stable trajectories of risk over 2 years in the CHR population. They used functioning and clinical symptoms. And what to me was most interesting about this, and this is just one example looking at a global assessment of functioning, there were these 3 trajectory groups identified, and they replicated the initial 3 groups from Naples 2 in the validation set across functioning and multiple symptom domains. And what of course is always a factor here when we look at these studies is what other significant predictors are there or confounding variables. And so in this analysis, age was significantly different between groups. So always thinking about mediators and moderators throughout this research. The other thing that's happened is we've begun to say, how do we combine these different clinical risk factors, genetic risk factors, other types of risks to estimate the psychosis, the risk for development of psychosis. And I've just pulled out a couple of examples. There are more. Many people on the phone whose work this is in the meeting today can talk more elegantly about the results of this. But we're not only seeing utility of these to be replicated in within one lab, we're seeing these markers be replicated across different labs and different data sets and used to predict psychosis risk over time. So really, really elegant and interesting and robust research that's leading us to the place we are now. Another critical factor that Dr. Heinzen was just talking about is outcomes. We talk about this in neurodevelopmental research, that while we're trying to find treatments that can be better for people in the future, causal factors that might contribute to developing novel therapies, we have a whole millions and millions of affected individuals, potentially affected individuals who are living right now, who need help right now. So what can we say about that? What we can say is that when we look at this, and this is just an example of a recent meta-analysis looking at 75 studies and outcomes over up to 36 months with different methodologies, looking at a whole host of different outcomes. And what they found was that outcomes are improving and can be improved with treatment. I think what I would say to this is also, though, that longer term outcomes are still not optimal. And it appears from at least this one meta-analysis that, and this could be a factor of the research, that we don't have longitudinal samples that go over many, many years at sample sizes that we could utilize more robustly. But what this suggests is that remission rates are peaking at about 24 months. That is, we don't see improvement in remission over time. So while we're seeing improvements, we're now tracking it. We're following outcomes over much longer periods of time in some of these sentinel centers, in networks, and trying to understand what predicts positive outcomes and what types of interventions can influence and potentially create remission. We've also vastly improved the approach that we're taking and the potential utility of biomarkers and biomarker methods. So there was a whole issue of schizophrenia research focused on development of biomarkers in clinical high risk. And so I think something that's really changed from how we were thinking about this 20 years ago is looking at the potential interesting mechanisms, the purpose of the biomarker, which we typically sometimes overlooked. We think a biomarker is going to be a diagnostic or a surrogate marker, and we don't take it as, we take it for granted that they all have the same purpose, but they actually can have quite different purposes. And also what potential biomarkers already we've been thinking about in the psychosis or psychosis spectrum, and at what point they would play a role in understanding whether or not, as Barnaby was talking about early in the day, differential outcome risk. Those people that convert, those people that are non-remitters, non-converters, or non-converter remitters. So really interesting research here that schizophrenia will continue to build on. We'll also tie these biomarkers and early translational phenotypes and knowledge to neurobiology and psychosis spectrum. So there were a couple of examples given earlier. One was mismatch negativity. This is a model that we can do, that we can use in preclinical assessment of genetic models, and also looking at outcomes with early treatments, and model that in the age models that we have of what's going on in the brain at the time that psychosis symptoms or clinical high-risk symptoms are emerging, what happens during the peripsychosis period, and what happens in chronification in schizophrenia. So as we begin to become more mature about our ability to assess these things in humans and then assess these in translational models and think about the brain areas that are affected, this will allow us to closer assess the pathways that are implicated in the chronification of psychosis and eventual potential diagnosis of schizophrenia. And of course, the science, including biomarkers, will better our understanding of developmental factors in the formation of schizophrenia, really from pathways to neurotransmitters to plasticity. And the more we think about the clinical signs and symptoms, the neurobiology, natural development and developmental factors, genetic risks, the more mature we're going to be in how we fine-tune and utilize these biomarkers to optimize clinical trial designs. So I wanted to really begin to close out my talk, talking a little bit about AM schizophrenia. Some of this has been talked about before, but what was to me was particularly compelling and interesting is we need better tools for predicting individual outcomes. We need to establish global research network. We were talking earlier about non-competitive research. That's a little bit of an oxymoron because there's always competition for research funding and competition of ideas, which promotes and hones healthy thinking. But the framework that we develop in these collaborative approaches that we take can help to establish a network that supports all of the good research that people are doing. We also need to set the stage for testing new, potentially preventive treatments. Now, this is something we were thinking about 20 years ago. You know, are we preventing a conversion to psychosis? Are we treating a syndrome right now? Are we preventing a chronification of psychosis to schizophrenia? Those things may require very different types of treatments and different types of approaches and different biomarkers. And then I think what is vital to all of us, for those of you who have been working in drug development for many years, you'll know that we've had many, many studies of, in schizophrenia looking at outcomes of novel medicines over the years. And very large databases where we can ask very interesting questions about outcome phenomenology. But those databases are not all accessible. They're not easily interrogated across the board. And I think we're moving in the direction that oncology has known and needed to move to 20 years ago. We're moving in that direction now, which is really, really exciting. I won't talk about Prescient, Pronet, or Predict. You've heard about these all day. But what I will say about this is that we need slightly different approaches, and we need organizational approaches to make sure that we are mature in the way that we're organizing our data, the way that we're thinking about analyzing that data. These data sets are massive. The types of imaging resources, blood-based biomarkers, genetic resources, takes real sophistication to manipulate, and organization of that data will help for that data to be able to be used by multiple different groups. And of course, developing larger and larger longitudinal observational studies that will have a more definitive data set that people can use to then build on for the next generation of studies and research. These tools will also then be able to be validated at a different level. And one thing that's particularly heartening about this to me is the approach of involving regulators early in the process and the discussion. We've done this in other forums, and what helps is it helps us to hone the scientific questions that we're asking and to meet the needs that regulators have in proving the science that will allow them to assist the field in developing novel drug development programs for novel therapies. This is the second time, I think it may even be the third time you've seen this slide today. Why am I showing it again? Well, why I'm showing it is because it speaks to the need for and the challenge that we have in research. When we have individuals who may have symptoms, who may be developing more symptoms, their involvement in research is very complex, and it takes a huge organization and a huge amount of thought that went into the design of this research program. How much time is being spent on the interviews? What's the sequence of events that's going to happen for each research? How each research institute is going to make sure that they best optimize that research process. This is something we do a lot in pharma. We do a lot of thinking about our protocols. How are we going to structure everything? How much time will it all take? Will it be too burdensome? Can we cut anything? And this was a multifaceted collaborative process amongst some of the best minds in the field, thinking about how all this would work. And we also implemented some very interesting and novel techniques. Phone digital diary with auditory recordings. Is this going to be something that's going to yield a biomarker? We don't know. But what we do know is that that type of information can yield very interesting factors that aren't easily picked up in other ways. Ecological momentary assessment, asking questions during the day, periodically a short number of questions, can be a very novel and interesting way to gather information. We have to do some standard interview questionnaires. These are critically important to us because they function as endpoints in our clinical trials and anchors. When they're taken, how they're spaced out, or what do we do? How do we impute missing data? All of these things become critical to us in our outcome assessment. And then, of course, when we look at more complex or more expensive measures like MRI and EEG, how do we integrate that into this process? So we're doing the best that we can to optimize the time that we have with people without overburdening them, allowing them to participate over time. So just really a remarkable body of work to get to this protocol and this set of measures. So I'd like to just wrap up saying, the future is to predict, preempt, and improve outcomes for all. We can't yet reliably predict those who will convert from clinical high risk to full psychosis or schizophrenia. We can't yet match novel therapeutics to early psychosis neurobiology, and we can't yet prevent conversion to psychosis at the individual or the subgroup level. But the AMP schizophrenia collaborative approach is going to lead us to what I think will be major revelations in the field and yield new science to inform psychosis risk and outcomes. And I'm thrilled to be a part of this. I know working with really wonderful partners, it's just a very exciting time. And if I, in 2003, had been told that we would be here with this science now, I would have been very, very happy. I would have felt like the work that we were doing back then really had a huge meaning, and I know many of you who were there at that time probably feel the same way. So just thrilling to see it happen. So I'm going to go back to my original schematic that I drew about where I think biomarkers are in 2022, and say, we're hopeful that the AMP program will lead to these biomarkers of stratification and enrichment and potential change measurement, and that ultimately that will lead us to novel therapeutics. So I'm happy to close there. I know I tried to make up a little bit of time for us, Linda, so we'd have plenty of time for the last talks. And I want to thank you all for listening to me today. Happy to take any questions you might have.
Thank you very much, Gan. That was really an inspiring talk, and it was a lovely perspective. We have a few minutes if people would like to ask any questions. It's really a nice synthesis of biomarkers and how it maps onto the science as it's progressed over time and where we are now. That was really fantastic, Anne.
Thank you.
Oh, question from Jerome Taylor.
Hi, Dr. Brady, and hi, Dr. Pandina. Great presentation. I am curious about how you think about, you mentioned cancer as being 20 years ahead of us, Dr. Pandina. Where are we compared to other fields of medicine? So like cardiology. So we still don't understand the mechanism really of hypertension, or like essential hypertension. So where are we compared to those, and are there innovations? So cancer is like super well funded, right? So I work at the Children's Hospital of Philadelphia, and everyone likes donating to cancer because it's something that can affect us all. Mental illness is harder. So how can we move from now to the future? So what do you feel like the next steps are to catch us up with oncology? Yeah.
Thank you. That's a great question. Well, I think there are a few things that are happening that are getting us in that direction. First, I think there's a recognition that if 1 in 5 people has a major mental illness, then every family is affected, right? There's no family that's unscathed. And much like we didn't used to talk about cancer at all, couldn't even say the word out loud without people being horrified. That's been true of talking about mental illness or psychiatric disorders. And I think the stigma that's associated with that has been something that still needs to be addressed, but at least it's being addressed and discussed. And I have hope for the next generation about that. I think I also, while I mentioned that we're behind cancer in oncology research, in a way we're right where we need to be and right where we should be, because the endpoints that we're looking at in the brain are much more complex. The biology is more complex and we don't understand it. And there are likely multiple pathways to all of the diseases, all of the disorders that we're talking about today. Schizophrenia being one of them, mood disorders, bipolar disorder. The phenomenology is all measured by subjective symptoms that are measured by experts rather than having an independent biology that we can test. In cardiology, we have these great surrogate biomarkers because we had hundreds of thousands of people right? So we knew, gee, blood pressure leads to, elevations of blood pressure lead to this diagnosis of hypertension, which leads to cardiovascular risk. We only did that through these large collaborative studies. And we did it because, of course, people were dying from it. I think now people realize the contribution of mental disorders to mortality is absolutely significant. So we know, that people who have cardiovascular risk and have a heart attack or heart surgery are much more likely to die if they have depression. Known fact, proven, very robustly. So I think that these initiatives, understanding that we also have a different developmental course for these disorders, and focusing on understanding that developmental course is really critical. I also think, we're going to end this day with, the patient perspective, right? We're going to end this day with people talking about that. That needs to be our lodestone. That needs to be our call to action, and we need to continue to listen to what people want. And in oncology and in cardiovascular disease, people say we're dying of this. We need, we need to be helped. We need to fund that because look, my father died at 60 and he could have lived to 80. And the same thing is happening in mental health now. And I think there's much more understanding that these are also, the disorders of the young, and they steal many people's adulthoods from them and divert them from achieving the outcomes that they'd like to achieve, even if they don't result in mortality. So it was a long-winded answer, but I appreciate the question and the ability to talk a little bit about that.
Thank you.
Well, thank you very much, Gahan, and thank you, Jerome, for that really great question.